extract treatment on survival rate, bacterial load, and organ failure in septicemic rats. Well, to refresh your memory, I would like to mention that sepsis and septic shock are one of the leading causes of mortality in intensive care units. Severe sepsis results in more than 200,000 annual fatalities, and the number of cases are projected to increase. Epidemiological studies of the incidence of sepsis indicate that between 11% and 27% of ICU admissions have severe sepsis, with mortality rates ranging from 20% to more than 50%. Further, I'd like to mention that sepsis is a severe systemic illness caused by invasion of pathogens to normally sterile parts of the body. This local infection causes a systemic response which leads to septic shock and death. It is a complex disease which causes oxidative stress and also imbalance between pro-inflammatory and anti-inflammatory processes. Free radical production is implicated in this process, both as a mechanism for direct cellular injury and in activation of intracellular signaling cascades within inflammatory cells, resulting in progression of the inflammatory responses. The clear evidence for oxidative stress in sepsis and the link with inflammatory gene expressions, including cyclooxygenase 2, namely COX-2, indissoluble nitric oxide synthase, namely INAS, and intracellular signaling pathways, including nuclear factor kappa B, NF-kappa B, and mitogen-activated protein kinase, namely MAPK pathways, have provided a foundation for interventions to either reduce oxidative stress, which could be through using antioxidant therapy, or inhibit transcriptional activation. Multiple organ dysfunction syndrome, one of the major causes of death from sepsis, is also resulted from oxidative stress and hyperinflammatory state. Some studies using antioxidants and anti-inflammatory agents found clinical benefits and survival improvement. Do you hear me? Ladies, gentlemen, madam?
Yes, we can hear. Please continue.
Well, pomegranate, the scientific name Ponica granatum, from the Ponicaceae family, a fruit with high content of polyphenols, have been focus of more recent studies because of its antioxidant and anti-inflammatory properties. The polyphenol content and antioxidant activity of pomegranate has been shown to be more and stronger than many potent antioxidants such as green tea. Its anti-inflammatory effects happen via inhibition of cell signaling pathways, including suppression of COX-2 and INAS expression, inhibition of activation of NF-kappa B, and inhibition of phosphorylation of MAPK proteins. The reported protective effects of polyphenols in sepsis includes improving mortality rate and controlling inflammatory and oxidative outbursts. However, there are some recent studies presenting no effect of polyphenols in similar conditions. To the best of our knowledge, the effect of pomegranate on sepsis has not been investigated extensively. According to high polyphenol content and antioxidative and anti-inflammatory effects of pomegranate, we initially hypothesized that pomegranate may protect against sepsis mortality. Please keep in mind, we initially hypothesized that pomegranate juice protect against sepsis, but our result turned out to be against this hypothesis. It was very interesting that I explained. The aims of this study were to evaluate the effect of high-dose pomegranate fruit liquid extract, namely POMEX, on oxidative and inflammatory response after disease induction in rat model of sepsis. As far as the pomegranate treatment and experimental design is concerned, pomegranate fruit juice extract, namely Pomex, was purchased from Pom Wonderful from LA, California. The Pomex is a liquid concentrated extract that is produced by extraction of the remaining fruit residue obtained after the first pressing for pomegranate juice and contains natural pomegranate polyphenol extract. Each 5 ml of extract contains 1,000 milligrams of pomegranate polyphenols. The rats were randomly, the design was that the rats were randomly divided to 3 groups of 8 each. The first group was sham group. The animals underwent the sham surgery without any other intervention. The CLP group, namely cecal ligation perforation surgery. The animals underwent CLP surgery without any other intervention. And Pomex group, the rats consumed 250 milligrams of pomegranate fruit extract per kilogram per day for 4 weeks before cecal ligation perforation surgery. The Pomex dose were selected according to previous studies which hypothesized to modulate oxidative stress and inflammatory outbursts in sepsis. Due to light-sensitive nature of Pomex, fresh Pomex was added to dark bottles containing drinking water on a daily basis. Well, as far as the sepsis model that we followed is concerned, I must explain that CLP surgery, namely cecal ligation perforation method, was used to induce sepsis in rats. Briefly, rats were anesthetized with intraperitoneal injection of ketamine, 80 to 100 milligrams per kilogram, and xylazine, 5 to 10 milligrams per kilogram. After deep anesthesia, rats were placed in a supine position and the abdominal wall area was shaved. A 3-centimeter incision was made in abdominal wall and cecum was exposed. As you can see in this slide, incision was made, cecum was exposed. And the cecum, the next step, the cecum was ligated at the middle to induce a moderate severity sepsis and was perforated twice distal to ligation placed with a 18G needle. Small droplets of feces were extruded from both holes. As you see in this photograph, that cecum was ligated and perforated, and 2 drops of feces was extruded in the abdominal wall in order to induce sepsis. And after relocating the cecum to the abdominal wall was repaired in 2 layers using a 4 sterile silk suture. Okay, as you see, yeah, the cecum was relocated and skin was repaired. And sham surgery, I explained. One group was sham surgery. In the sham surgery group, sham surgery was also made in control group with the same procedure except the ligation and perforation step. Without the sham surgery, the surgery was made, but ligation and perforation step was not done. All the animals were resuscitated by injecting pre-warmed normal saline, 37 degrees centigrade, 5 ml per 100 gram body weight, subcutaneously, and were kept under close observation in a warm room until recovery. All the animals had free access to food and water after recovery. Actually, first blood sampling was carried out just before CLP surgery, and subsequent samples were taken 24 hours after surgery. All the animals were sacrificed humanitarily to collect their blood, peritoneal neutrophils, and tissue samples. Blood was collected by cardiac puncture, and sera were separated and kept at -80 degrees centigrade before analysis. Peritoneal myeloperoxidase activity, tissue superoxide desmodase, catalase, and glutathione peroxidase activities were measured. Serum total antioxidant status, namely TAS and T-bars, thiobarbituric acid reactive species, were determined. Serum nitrate and nitrite and serum cytokine assays INES and COX-2 expression measurements were carried out. And finally, organ failure examinations were conducted through histological and biochemical methods on different tissues. A statistical analysis also was made by using SPSS software. Normality of data distribution was tested by one sample Kolmogorov-Smirnov test. Since the data were not normally distributed, the difference between groups was studied by Mann-Whitney-U non-parametric tests. As far as the results are concerned, it is interesting I mentioned that effect of pomegranate polyphenol, effect of these treatments upon the myeloperoxidase activity. That's a very important issue that, as I said, our result was against our initial hypothesis. You see, in the myeloperoxidase activity is one of the important enzyme activity that act against the infection at the beginning of the infection. And unfortunately, we suppressed this myeloperoxidase activity by hypo-inflammatory effect of the pomegranate juice. That was detrimental. You see, in the CLP group and POMEX group, the group that received the pomegranate juice, the activity of myeloperoxidase activity dramatically suppressed and reduced compared to the sham group. That was detrimental. That was detrimental. And as far as the serum total antioxidant capacity is concerned, our result that showed that when the group used consumed the pomegranate juice without surgery, of course, when we compare that to the group that does not receive the pomegranate juice, without surgery, the level of the antioxidant capacity is statistically significant compared to those that non-consuming POMEX. It means that pomegranate juice administration increases total antioxidant capacity. But look at here, when the rats undergone the surgery, you see the total antioxidant capacity of both CLP group and POMEX group, those that received pomegranate juice, significantly reduced compared to the sham group. That is detrimental. That is detrimental. And with regard to thiobarbituric acid reactive species, that is indicative of the tissue damage. You see, in the CLP group and in the POMEX group, the group that received pomegranate group, the level of the T-bars significantly is higher than sham group. That is detrimental. This against, is life-threatening. But in the lungs, there is no significant differences among these three groups with regard to the T-bars. As far as the antioxidative enzymes are concerned, namely superoxide desmutase, in the three groups, namely CLP and POMEX and sham group, there is no statistical significance with regard to the superoxide desmutase in the liver. And in the lung, also, there is no significant difference. But with regard to the catalase activity, another antioxidative enzymes, as you see, there is a significant increase in the POMEX group compared to the CLP group. And in the lung, there is no significant differences among three groups. With regard to another antioxidative enzyme, namely glutathione peroxidase, again, there is significant increase of glutathione peroxidase activity in the POMEX group compared to the CLP group, and also compared to the sham group, in the liver. But in the lung, again, there is no significant differences. With regard to nitrate and nitrite, that's very important. Nitrate and nitrite is a precursor of, is the nitric oxides, and in the sepsis, damaging the life, is life-threatening. Increase of nitric oxide is life-threatening. But on the other hand, nitric oxide is protective of the cardiac tissue. Is the cardiovascular, in the cardiovascular diseases is protective, but for the septic shock is detrimental. You see, when we administer the pomegranate juice, you see there is significant increase in nitrate and nitrite, that is nitric oxide species, both in the CLP group and in the pomex group, compared to the sham group, that is almost very little activity, very little concentration, or even negligible. So in the septic shock, by administering pomegranate juice, nitric oxide concentrations is increasing dramatically. That is detrimental. With regard to cytokines, again, TNF-alpha, you see that by administration of the pomegranate juice, the level of the TNF-alpha increases dramatically, and also in the CLP group compared to the sham group, that is negligible. The level of the TNF-alpha in the sham group is negligible, but by administration of the pomegranate juice, it increases dramatically and is statistically significant. That's detrimental. And also serum interleukin-6, again, by administration of the pomegranate juice, increases dramatically, both in the pomex group and also in the CLP group compared to the sham. And also interleukin-10 increases again significantly compared to the sham group. With regard to INS expression and also COX-2 expression, both inducible nitric oxide synthesis, inducible nitric oxide synthesis that produces nitric oxide. As I said, the inducible nitric oxide synthesis in septic shock increases significantly and produces a lot of nitric oxide. You see, compared to the sham group, the level of the activity of INS dramatically is higher than sham group. That's detrimental. And also COX-2, cyclooxygenase 2, again, expression of this COX-2 significantly is higher than sham group. That's detrimental. So, and also with regard to effect of the pomegranate juice upon organ failure, we use two types of parameters, one biochemical, one histological. We use the parameters indicative of the organ failure, namely BUN, creatinine, SGOT, SGPT, LDH, and bilirubin concentration. We didn't find any significant difference among these three groups, CLB group, pomex, and sham group. There was no significant differences. With regard to histological examination, again, when we examine the liver, lung, and kidney tissues in these three treatments, sham, pomex, and CLP, there were no significant differences with regard to the histological changes. Well, if I want to put these findings, I must say that interpretation of these findings need understanding of sepsis pathogenesis, and mechanism through which other polyphenols affect sepsis outcome. After bacterial recognition, inflammatory response is stimulated through various signaling pathways. Although hyperinflammation has been shown to cause multiple organ disease syndrome and mortality, but proper inflammatory response is needed to control the infection and efficient function of immune system. So the first defense of body against infection is myeloperoxidase activity from the lymphocytes, and by administration of hyper doses of pomegranate, we suppress this first level of the defense. That's detrimental. Although hyperinflammation has been shown to cause multiple organ disorders, syndromes, and mortality, but proper inflammatory response is needed to control the infection and efficient function of immune system. An extreme hypoinflammatory response, that's what happened in our research. An extreme hypoinflammatory response that's caused by administration of the pomegranate juice, which leads to lack of inflammatory cytokines and activation of adaptive immunity, will cause insufficient immune system response, which subsequently leads to bacterial infection spreading all over the body. Indeed, the mechanism of septic shock must be considered too. The vasodilation and myocardial dysfunction occurring at late sepsis is responsible for shock progression with nitric oxide produced by ions, has a major role in this context. So increasing the nitric oxide levels by inducible nitric oxide synthase is detrimental in septic shock. Other points to consider is how polyphenols prevent or treat sepsis. It seems that anti-inflammatory properties of polyphenols are the major mechanism of their beneficial effects. In studies reported so far, polyphenols have been shown to inhibit multiple organ dysfunction through the modulation of primary inflammatory response in sepsis. That's true, which is accomplished by attenuation of local and systemic production of NF-kappa B dependent inflammatory mediators, including tumor necrosis factor alpha, COX-2, and intercellular adhesion molecule 1, namely ICAM1. Suppression, that's important too. Suppression of INS expression by polyphenols is another mechanism by which they improve sepsis outcomes. However, the safety and probable toxicity of polyphenols should not be forgotten. The harmful effects of polyphenols include potential for causing dose-independent or dependent adverse effects, such as toxicity or endocrine system disrupting effects of certain polyphenols. Considering above information, the results of our study put forward two distinct hypotheses to understand the cause of these aggravating effects. First, the detrimental effect may be due to anti-inflammatory effect of pomegranates. Since the bacterial load in treatment group is significantly higher compared to sham group, by suppressing the initial inflammatory effect, you provide the situation, the atmosphere for bacterial load and severe infection. It seems that POMEX may cause an extreme hypoinflammatory response, which leads to higher bacterial load. That's what caused in our experiment. By administration of POMEX and creation of hypoinflammatory response, the higher bacterial load was taking place, and severe infection and sepsis was taking place, and thereby increased mortality rate. Although the beneficial and anti-inflammatory effect of similar dose of pomegranate extract was shown in other studies of chronic inflammatory state. Please just make difference between chronic situation and acute situation. The septic shock is an acute situation, and maybe the high dose of pomegranate is beneficial in the chronic inflammatory state, but not in the septic shock that is acute, as acute status. This dose of Pomex seems to be unsuitable for sepsis, an acute inflammatory condition. It is yet to be determined whether other doses of Pomex will improve the condition or cause adverse effects in sepsis, independent of dose. The second hypothesis is the effect of pomegranate on INS and endothelial nitric oxide synthase. ENOS is two different. INS is inducible nitric oxide synthase that is detrimental for septic shock. But we have endothelial nitric oxide synthase, ENOS, that is beneficial for cardiovascular diseases. Although pomegranate has been shown to suppress INS expression, long-term consumption of pomegranate extract was demonstrated to increase arterial ENOS expression and plasma nitrate and nitrite levels. We hypothesize that this background high nitric oxide may make the animals prone to septic shock. Yeah. So increasing nitric oxide prone animals to septic shock and death. Besides, pro-inflammatory role of ENOS was shown in endotoxic shock models. Further studies are needed to test all these hypotheses. Incomplete levels, so I conclude, incomplete levels of inflammatory responses may prone the organism to outgrowth of infectious agents and consequently development of more severe diseases. That's very important issue, that neutrophils are key effectors of the innate immune response, and their migration to infectious focus is critical for the rapid eradication of bacterial pathogens. That was a very important issue. When we administered the pomegranate, we suppress neutrophil and we suppress the myeloperoxidase activity that is secreted from neutrophils. And it caused the outgrowth of the bacteria. Although their hyperactivation may contribute to the development of multiple organ failure in sepsis, their insufficient activation at the beginning of disease establishment also may aggravate the disease and enhance its severity. Early enhanced local neutrophil recruitment accomplished by higher neutrophil myeloperoxidase activity in the animal model of sepsis was shown to improve bacterial clearance and animal survival. We suppress that, unfortunately. Myeloperoxidase is a neutrophil enzyme which produces free radicals to combat with the bacterial pathogens, and its deficiency was shown to impair survival in animal model of sepsis. Considering the fact that proper immune responses of neutrophils and myeloperoxidase activity are needed to cope with infection, an immediate question rises on how the extent of sepsis and its subsequent complications are affected by high doses of pomegranate extracts. Pomex consumption prior to sepsis induction suppressed the vital function of neutrophils in early hours of sepsis induction, thus resulted in higher oxidative stress after CLP induction. The result may suggest that despite its anti-inflammatory action in chronic conditions, pomegranate may not work properly in rapid progressing conditions like sepsis. So I suggest that we should not fall in the pitfall of the antioxidants like pomegranate or other antioxidant materials. To our best knowledge, this is the first study investigating the effect of long-term consumption of high-dose pomegranate extract on peritoneal neutrophil myeloperoxidase in CLP model of sepsis. However, our study had some limitations. We investigated a simple dose of Pomex. We used a simple dose. Lack of dose response experimental group is the major limitation. Thus, the effect of other doses is yet to be determined. In conclusion, our study demonstrates that pomegranate extract could increase mortality rate via increasing peritoneal cavity bacterial load in CLP sepsis model. High-dose Pomex supplementation before sepsis induction resulted in impaired activity of peritoneal neutrophil myeloperoxidase activity, a key enzyme for controlling infection at first hours of sepsis initiation, and consequent higher oxidative stress as judged by decreased serum total antioxidant status and increased level of T-bars levels. thiobarbituric acid reactive species that is indicative of the tissue damage. These findings indicate that caution should be made in using high-dose pomegranate products. The main message of current study is that such useful compounds as antioxidants, including pomegranate juice, which have beneficial effect on general health status, may have detrimental effects if misused or used in high doses. Thank you very much for your patience.